Hello everyone, this is part 6 of chapter 6. In this part, we have to discuss remaining part of the chapter 6. That is, here we have to deal secondary growth in dicot stem and dicot root, what happens inside the stele and outside the stele in this, and then we have to see unrolling formation, what are the differences between sapwood and heartwood, and because this is the last chapter, last part of the chapter, therefore, we have to see NCERT exemplar and previous year examinations questions. This is part 6, chapter 6. Anatomy of flowering plant. In this part, we have to discuss secondary growth. You are familiar with primary growth. Primary growth in root and stem is because of apical meristem, that is shoot, and that is root meristem, and this growth is known as primary growth. In dicot stem and dicot root, secondary growth is also found. What we mean by when the increase is there in girth, that is diameter increases, thickness increases, this is called secondary growth, and this secondary growth is because of lateral meristem, that is vascular cambium and cork cambium. Secondary growth we have to see first in dicot stem, then we'll proceed secondary growth in monocot, that is in dicot root. Secondary growth in dicot stem, as you are familiar that in dicot stem vascular bundles are, these are conjoint, these are arranged in a ring, and these are open, that is the cambium is found here in between. That is, bundles are open as they have cambium, and this cambium which is found in between, that is primary phloem and primary xylem. This cambium is known as intrafascicular cambium. So here this is intrafascicular cambium. Now at the time of secondary growth, what happens now? That is, these are the vascular bundle which are arranged in ring, and this is the intrafascicular cambium, that is in between the two vascular bundles, that is the medullary rays are present. These are the medullary rays here. The cells of the medullary rays which are exactly in front of, that is intrafascicular cambium, they also become meristematic. That is, by de-differentiation they will become meristematic, and this cambium formed, this is known as interfascicular cambium, while this cambium, this one is intrafascicular cambium, and both together they will make, that is cambium ring. A circular cambium ring is formed. You can see the diagram here, that is, this one is primary phloem, this one is primary xylem, this is intrafascicular cambium, and now in between the vascular bundle, this one is interfascicular cambium, this is intra, and this one is inter, and in that way a circular cambium ring is formed. Now we have to see what happens after cambium ring is formed. Activity of cambium ring. Cambium ring that becomes active, and it starts to cut off new cells, outer side and inner side. You can think in that way, that this is the cambium ring here, that this is the outer side, that this is the center here. It will cut the cells towards inside and towards outer side. The cells which are cut towards inside, ultimately they will differentiate as secondary xylem, and the cells which are cut towards outer side, they will be differentiated as secondary phloem. This is secondary xylem towards center, and this one is secondary phloem towards, that is outer side, periphery. What happens? The cambium is generally, you see, very important point, more active towards inner side, more tissue is added towards inner side as compared to the secondary phloem. More tissue is added towards inner side. Amount of, therefore, secondary xylem produced is more than the secondary phloem. Is it? Come to the next point, primary and secondary phloem, that is, they gradually get crushed. You know, if this is the vascular bundle here, that was the primary xylem here, and that was the primary phloem. You know, if that was the vascular bundle, and this is the vascular cambium in between the vascular bundle, inside the vascular bundle, then what will happen? Because of formation of secondary xylem, this primary phloem, that will be shifted towards center, and ultimately, that is because this is made of hard tissue, will not be crushed, that will remain till last. But here the primary phloem, because these are made of delicate cells, that will be crushed in between the secondary phloem, and that is endodermis. You see, primary and secondary phloem gradually crushed due to, that is what, continuous formation, accumulation of secondary xylem. But what happens now? Primary xylem, however, remain more or less intact. This is primary xylem, because xylem is made up of hard-walled tissue, thick-walled tissue that remain intact, and around the center that will be there. At some places, cambium that makes narrow band of parenchyma. You see here, this is the cambium ring. Here at this place, it is making secondary phloem and secondary xylem, but at certain places, the cells added towards outer and inside, they do not differentiate at secondary xylem or secondary phloem. They remain parenchymatous, and that is secondary medullary ray. That will make secondary medullary ray. Now you can see here that this is, what is this? This is the cambium ring. You are seeing cambium ring is this one. That is, this is the secondary xylem, which is more, that is amount, while outside that will be secondary phloem. This is the cambium ring here, and in between this is the medullary rays, secondary medullary rays, and this one is the cambium ring. Is it clear? You can see once more here that this is cambium ring. That is here the tissue formed. That will be what? That will be the secondary xylem. Is it? Here outside that will be that is secondary phloem. Here that will be, that is primary xylem. Here that will be primary phloem. And in between, that is somewhere, these are the medullary rays which do not differentiate as secondary xylem and secondary phloem. Is it clear? The same thing we have shown here. That is, this is, what is this? This is the primary xylem. This one is primary xylem which remain intact, and here you can see the tissues the same here. Is it right? Come to the next now. That is annual ring formation. What? Annual ring, also known as growth ring, and this is because of spring wood, and that is autumn wood. How this will be formed? You see, activity of the cambium is under the control of many physiological and environmental factors, especially in temperate region where the seasons are formed. You know, seasons will be, that may be favorable season, and that may be the unfavorable season. For example, that is spring season is the favorable one. For example, the winter season, that will be the unfavorable one. What happens now in spring season? The activity of cambium ring will be more active. That will be more active as compared to the winter season. When it will be more active, it will make, that is, the amount of xylem and amount of phloem formed, that will be much more. You can see here, that is temperate region. Let this is Shimla, Nainital, that is Mussoorie. The climatic conditions are not uniform. The seasons are there. That the spring season, in spring season what will be? Cambium is very active, and it produces a large number of xylem element, and having the vessels formed in spring season, they will have, that is comparatively larger diameter. While that will be formed there in winter season, that will be of narrow diameter. If you think in that way, that this is the narrow diameter, and here, that is when the season was spring one, the xylem was formed with wide diameter. You see here, that is the wood formed during this season will be known as spring wood or early wood. That will be spring wood or early wood, that is in spring season, and here this wood will be known as, that is late wood, or will be known as, that is autumn wood. Then what in by spring? That is annual ring now. That is spring wood. You see, it is lighter in color, that has lower density, where the autumn wood is darker and has high density. The two kinds of wood that appears as alternate concentric rings, it constitutes the annual ring. Annual rings, that is seen in cut stem, and by this we can estimate the age of the tree. Growth rings, that is, are not distinct. You see, where the place where the seasons are not formed or not distinct. For example, that is tropical areas, or this is the seashore, near seashore in our country. That is Mumbai is there, Calcutta is there, or Chennai is there. So here the seasons are not formed. The seasons or climatic conditions are almost same throughout. What happens? Therefore, the cambium activity will not be changed. It almost remains same throughout the year, and therefore the annual rings are not formed here in such places. More clear annual rings are formed. This is the important one. Deciduous forest, because deciduous forest, they have favorable and unfavorable season. In desert, annual rings are less distinct. Why? Because the season is almost same throughout the year. Annual rings are band of secondary xylem, and that is xylem rays. It's not because of phloem, that is annual rings are only because of that is secondary xylem. Is it clear? Come to the next now. We have to see now, that is the next part, that is we have to see heartwood and sapwood, what we mean by. You must have seen that is a log wood like this, that is, you see, this is the central part which is darker in color like this, and this is the peripheral part which is comparatively light in color. So this wood which is that is in the center, that is known as that is heartwood, and which is found at the periphery, that is known as sapwood. That is, we can differentiate heartwood and sapwood, that it makes that is central major portion of any dicot stem, while the peripheral portion is made up of sapwood. This is darker in color, it is lighter in color. Why it is darker? Because here we have deposition of organic compounds like tannins, resins, oils, gums and aromatic substances and some essential oils, and because of this wood will become more durable, more resistant to the attack of microorganisms. That's why it is much more, it is costly comparatively, because this is more resistant and it provides mechanical support to the plant. On the other side, that is, this is sapwood is light in color, because all these substances, these are absent here, that is, these features are absent. And here important thing is that this sapwood, very important point, this is that is physiologically, this one is active. Physiologically active, what we mean by that is water will be transported only through sapwood. Otherwise this heartwood, this is dead one, it doesn't have any physiological role. Is it clear? Come to the next now, that was inside the steel. Now you have to see what happens outside the steel. You see in this, that is the section of a dicot stem. You have seen here that the vascular cambium ring was formed, and because of this, that is secondary xylem was being added, that is secondary phloem was added outer side. What happens? Because of the addition of this secondary tissues formed by that is vascular cambium, what will be? The pressure will be generated on the tissue outside the endodermis. You see, by addition of this endodermis and all this will burst. As the stem continue to increase in girth, due to the activity of vascular cambium, the outer cortical layers, they will rupture here. Because the pressure generated from inside, the epidermal cells, these are being broken, and that's why there is need to replace, that is to provide protective cell layer. What happens now? That is sooner, that is what will be another meristematic tissue called cork cambium or phellogen. It will develop here. It will develop in the cortical region. That is usually it develops from that is outer cortical layer. This phellogen is a couple of layer thick and is made up of narrow, thin-walled, nearly rectangular cells. You see here, I can enlarge it here, that this is the cork cambium formed, that is just to give protective layer. Now this cork cambium, also known as phellogen, it will cut cells outer side and inner side both. Outer side cells, they will become the cells like this, and these cells, these are suberized, and that will make the cork, or also known as that is phloem. This is cork cambium. This one is that is cork cambium. This is cork, and inside the parent parenchymatous cells will be added, which is known as secondary tissue or phelloderm. This is secondary tissue. The other name to the cork cambium, that is phellogen. Phellogen this is, while that is the phellogen cut off cells on both sides, as I told you. Outer cell differentiate as cork or that is phloem, and the cells added towards interside, these are parenchymatous thin-walled cells, also known as secondary cortex or phelloderm. Right? Now the cork, you see, as it is suberized, the cells are suberized, it doesn't allow that is movement of water, it is impervious to water. The cell of the secondary cortex, these are parenchymatous, I told you now, and that is all these three together, phloem, phellogen and phelloderm, collectively known as that is periderm. Due to activity of cork cambium, what happens now? That is the pressure build up and the remaining layer of the peripheral phellogen, that is ultimately these layers they die, and that is they will be given off. You see what we mean by park, important. Bark, what in by? This is a non-technical term. You see, this is the vascular cambium, for example. All the tissue outside the vascular cambium, that is known as bark. It refers to all tissue exterior to vascular cambium. Can you recall what will be there in vascular cambium now? Yes, this is vascular cambium. You can see this is secondary phloem. That is primary phloem. That may be the cortex here. Outside the cortex, that is secondary cortex, right? Outside the secondary cortex, that may be cork cambium. This is cork cambium. Outside the cork cambium, that may be the cork. This is cork, that is cork cambium, and that will be the epidermis. So all these tissue outside the vascular cambium, these are known as what? That is bark. Bark refer to number of tissues. I told you it includes periderm and all this. Just now I told you. Bark that is formed early in the season is called early or soft bark, and towards the end of season that is known as hard bark, or that is late bark. Okay. Now various kind of the cells that constitute the bark you have seen right now. It includes the phloem, and you can count. This is vascular cambium. Again, this is secondary phloem, primary phloem, cortex, secondary cortex, and all that will be included here in that. Is it right? Come to the next now. We have to see what we mean by that is lenticell. You see here that this is the cork cambium. I told you right now cork cambium. Cork cambium, this is made of rectangular cell, meristematic this one is, that is cork cambium. Cork cambium outer side that will make the cells which are suberized cell wall, this one is, and these are impervious to water. They won't allow water to go outside. But at certain places what happens, that is the cork cambium doesn't make the cork. Here it will make the thin-walled cells like this, and these thin-walled cells are known as complementary cells. This one is cork cambium, is it? This one is the cork, and these are the complementary cell. Because the formation of this, the epidermis that will be raised just like lens-shaped. That's why these area are known as lenticels. Come to this point, at certain region, phallogen cut off closely arranged parenchymatous cell on the outer side instead of cork cell. These are the cork cell. In this region, the cork cell will not be formed. These parenchymatous cells, they rupture the epidermis and they form a lens-shaped opening called lenticels. What will happen through lenticell? You know, that is here, the gases will be exchanged, the CO2 or that is oxygen that will go in, that is permit exchange of the gases, right? Internal tissue and atmosphere, and lenticels, it also that is permits transpiration of water. Lenticels occurs in most of the woody trees, permit exchange of gases. That's why these are known as that is breathing pore, right? Lenticels are present all over the plant body. That is even in fruit these are found, and that is the mean for gaseous exchange. You can see the diagram here. This is the secondary cortex, is it? This is the cork cambium. You can see that this tissue is, this is the cork cambium 1, this is cork cambium. Inside this is secondary cortex. Here these are the complementary cell. This is the ruptured epidermis, and this is the lenticell through which the gases will be exchanged. Is it right? Come to the next now. We have to see secondary growth in dicot root. You know, in dicot root, vascular bundles are little different from that of stem. If you see, that is in case of dicot root, that this is the endodermis we have here. That is endodermis. I will just recall that the vascular bundles, these are radial vascular bundle. That is, this one is the primary xylem, you know, and this one is that is primary phloem. This is primary xylem, and this one is primary phloem. Important point here is that this type of vascular bundle is radial and exarch. What is very, very important one, that is in dicot root, here the cambium is not present since beginning. You know, in case of stem, in between the primary xylem and primary phloem, the cambium was present since beginning. That cambium that is intrafascicular cambium. But here in this case, that is the cambium in between the band of xylem and phloem is not present here. Very important one. What happens now? That is this is the tissue in between this primary xylem and primary phloem. This tissue is known as conjunctive tissue. This tissue is conjunctive tissue. That is, the cells of this will become meristematic one, and that will make the intravesicular cambium in between the primary phloem and primary xylem. Is it right? Portion. Is it clear? This is the cambium formed right now at the time of secondary growth. One more thing will happen, that is the cells of pericycle, which is just in front of this is protoxylem. That is what will happen here. That is, the cells here, they will become meristematic one, and that they will make a band like this. In that way, a vascular cambium ring will be formed, which is not circular. This cambium ring, that will be the wavy like this. Are you getting my point? This wavy cambium ring will be formed like this, and this wavy cambium ring will be formed by union of inter and intravesicular cambium. So what is very important one, the cambium ring in case of root is exclusively, that is, secondary meristem. It is exclusively secondary meristem. Either this intra or inter will be formed at the time of secondary growth only. Cambium ring in the same way will produce secondary xylem on inner side and secondary phloem outside, just like a stem. The root doesn't have annual ring. Little later you see that this wavy cambium ring, that will become, that is, circular, that is cambium ring, and now it will start to make secondary tissues, outer side secondary phloem, inside that is secondary xylem. Here, because the annual rings are not formed, because the climatic changes are not observed inside the soil, from outer part of the pericycle, that is core cambium, very important one. Here in the same way, that is, this is the inside the stele. What happen outside the stele? That here, this pericycle that will make the core cambium, and in the same way, core cambium will generate, that is tissue outer side, that is cork, as I told you in case of stem, that this is core cambium in root also. Outer side that cork will be formed, and inside, that is the secondary cortex will be formed. Is it right? So this is about secondary growth in, that is dicot root. You can see here, this is the vascular bundle. That the vascular bundle, I told you, vascular bundle, these are radial, and the xylem, that one is, that is exarch one. What happens now? You are seeing that this is a wavy cambium ring is formed here. Wavy, that is cambium ring is formed here, and this wavy cambium ring, you see, now it has become circular. This is circular cambium ring, and inside it is going to make, these are the xylem, and outside that will make the secondary phloem. Is it right? Now this is the section of, that is secondary growth in a dicot. What will be here? That is epidermis, or that is peridermis. Usually that will be formed similar to the stem. That will be the, that is the structure like this. Is it clear? Should we proceed to the next now? Yes. Now we have to see, that is some NCERT exemplars. Good questions. Few questions we will discuss. Transverse section of the stem is strained. Very important one. Actually, this is general knowledge question. You see, in case of plant, the hard tissue where we have, that is lignin, that may be stained with the help of stain known as safranin. That is safranin, is it? While the, that is, this is called, that is safranin, and the thin wall tissue is being, that is stained with the help of, that is green. Okay. The question here is, that is stained with safranin section, then fast green uses. Okay, you can see double staining preparation we have done. What would be the color of the stained xylem and phloem? You see, xylem is made up of lignified tissue, that's why that will be red in color, and phloem is made of parenchyma tissue, that will be green in color. Match the following, choose the correct option. You can go through this question. That is, the right option will be here B. A, the right answer is, that is third, dividing cell. B, that will be photosynthesis like this. Can you compare at your own? Okay, come to the next now. Identify the simple tissue from among the following one. Xylem, that is complex one. Phloem, complex one. Epidermis, this is epidermal tissue system. Simple tissue, that is, this is parenchyma. Cell of the tissue that is living and shows angular and ball thickening. They also provide mechanical support. You know, living and, that is, the mechanical support is provided by collenchyma. Right? Epidermis of root is equivalent to Yes, epidermis of stem. In case of epidermis, in case of stem, and in case of leaf, the outermost layer we used to call that is epidermis, while the same layer in case of root is known as epiblemma. Phloem, phloem respectively, these represent cork cambium and cork, I told you right now. Some questions we can discuss which have come in, that is previous year examination. Interfascicular cambium that develops from, in case of that is, question is related to dicot stem, it develops from medullary rays. Age of the tree can be counted by counting number of annual ring. Column of water within xylem vessel the torti, how does it go? That is, that doesn't break because tensile strength of the water is there. Vascular cambium normally gives rise, that is secondary xylem. Is it? One more question we may see here, that is made up of dead cell, that is collenchyma living cell, phloem living cell, it also having living cell. The phloem, that is cork, is made up of exclusively, that is dead cell. Come to the last question here, you can do, identify the incorrect statement. 𝕍𝕀𝔻𝔻 is innermost secondary xylem and it is light. No, 𝕍𝕀𝔻𝔻 is, that is at the periphery, and this is, that is lighter in color, definitely lighter in color, but it is not the innermost, that is found at the periphery. Is it right? This is about the, that is last part, part 6 of chapter 6, that is anatomy of flowering plant. Okay, thank you.